Okay, thank you so much, guys, for coming. So today we are covering immune system 2, which is the last lecture for midterm 2, actually. Last time you did immune system 1, where you talked about the innate immune systems, where you pretty much talked about the natural defenses that are not specific, such as what are some of the examples for that, for innate mechanisms of defense? It's too early. Like macrophages, they engulf any kind of bacteria, right? Your skin is your first line of defense. So today we'll talk about adaptive mechanisms where this will be attacking a specific microorganism. So we'll talk about antibody formation, we'll talk about cytotoxic T cells that kill specific, foreign microorganisms. All right? So adaptive suggests something that kind of adapts to a certain, microorganisms. It will be made specifically for that particular microorganism. So it will adapt to that. So it will recognize a specific, that's the important part. These are the differences between innate and adaptive mechanisms. So the first time the body is exposed to a microorganism, it is naive, it doesn't know about that microorganism, so we'll start talking about primary response. And then after that, the body will build up memory cells, like memory B cells, memory T cells, memory helper cells, that will then recognize it the second time the body is infected, and then it will launch a more robust secondary response, right? So what this system does, it will immobilize, neutralize, destroy any foreign, microorganisms, all right? And what it will amplify inflammation, which is one of those innate mechanisms, right? It will amplify inflammation and also activates complement. Remember what complement is? What is complement? From immune system 1, because it's part of the what is complement? It's a group of what? Carbohydrates, proteins, lipids? Proteins, yeah. It's a group of proteins that will lyse cells by inserting themselves in the cell membrane to make a pore, and then the cell pretty much gets out. Okay, so complement is active proteins that will destroy foreign microorganisms. Proteins. All right, so that's that. So adaptive system is specific. It creates memory cells. Memory cells are responsible for launching a robust secondary response. All right. Secondary response is more robust because of these memory cells that will recognize that specific microorganism. All right, and it's systemic. Okay. It is throughout the whole system, okay? And these cells are, moving in blood that are killing these microorganisms. So it's systemic, it's specific, and it has memory cells. So this is the major difference between adaptive and innate. Adaptive has memory, okay? All right, and so there's two types where you have antibody-mediated system where you produce plasma cells from B cells, and then you produce a whole bunch of antibodies that are specific to that particular microorganism. So this is antibodies, all right? And then you have another one where you are using cytotoxic T cells to kill that particular, so it's cell-mediated, it's cellular, okay? It's cell-mediated, all right? And this will be the lymphocytes, like the T cells, okay? The T cells mostly involved in killing viruses. So they will kill cells that are infected with the virus, all right? So we'll start to talk about the major histocompatibility complex, which is a protein that is used to mount the antigen on the surface of cells. There's two kinds, 1 and 2, okay? And 1 is the one that is involved in cell-mediated immunity, major histocompatibility complex 1. Okay? Because that one, whenever an antigen is presented or is exhibited on the cell surface by MHC1, it shows that that cell is infected. So that cell is going to be killed. You see what I'm saying? And then major histocompatibility complex 2, I'm going to try and introduce this now so that when you get to it, you can have an idea between this, the way the immune system differentiates between MHC1 and MHC2. Usually, professional antigen-presenting cells like B cells, dendritic cells, macrophages, okay, when they engulf an external microorganism and they process it, they will present it on the cell surface as MHC2, okay? And when MHC2 is exhibiting an antigen on a cell, that cell is not killed. That cell will be part of the professional presenting cells that will then create other, molecules like cytokines that will kill. So that cell is not marked for. So that's how the immune system knows whether to kill that particular cell or to leave it alone because it's part of the immune system cells. Okay, so it will stimulate production of cytotoxic T cells or helper T cells and things like that, and antibodies. All right, so I'll introduce this again later on. Okay, but I wanted to bring it now so that you kind of have an appreciation between the two. So we know what antigens are. They are, molecules, protein chains. They are actually big. They even go up to 10,000 molecular weight, and they will stimulate a response from the immune system. All right, they will stimulate the adaptive immune system to respond. So any antigen is that molecule. It's part of an organism. All right, and so what the body does is we try to distinguish between self-antigens, because our body cells have antigens as well. Okay, and then so the body has to distinguish between those two and also foreign antigens. Sometimes when that process has something wrong goes with it, then you start to have immunological diseases, autoimmune disease, where you are attacking your own cells because you cannot distinguish your self antigens from foreign antigens. Okay? So the adaptive immune system is based on the ability of the body to determine which are foreign antigens and which are self-antigens, so that it doesn't attack itself. Okay? Because by definition, anything that can excite the immune system, is an antigen to respond. So, like we said, the B cells are responsible for forming antibodies. Antibodies are specific for a particular microorganism, all right? And B cells will first of all differentiate into what? Plasma cells, right? B cells will differentiate to plasma cells. Plasma cells are the ones that produce antibodies, okay? Not the B cell, all right? So B cells are responsible for the antibody-mediated immunity because they are the ones that differentiate into plasma cells, all right? And then the T cells are the ones that are responsible for the cell-mediated immunity because you have the cytotoxic T cells that kill infected cells, all right? So just know the differences between those two. The B cells, the ones that mature where? Where do B cells mature? Where do B cells mature? B cells? B for? Bone. So with them, so the mature T cells mature in the thymus. Yeah. All right. So just keep that in mind, okay? Because when we start to talk about tolerance, we'll talk about the two types of tolerance. Okay, so this is what it is, right? B cells, we know they are produced in bone marrow because B for bone and T for thymus. They mature in the thymus. but all the cells originate where? In this, from the stem cells in the red bone marrow, right? Okay, so when these B cells and T cells are being made, okay, positive selection is selecting, B and T cells to survive that attack foreign antigens. All right. So that's positive selection. You will select for the ones that will attack foreign antigens, right? Because you want that. You don't want, cells to survive, immune cells to survive that will attack your own antigens. All right. So negative selection will eliminate any lymphocytes that will attack self-antigens. It's selecting against negative, okay? Positive, you will select for those that will attack foreign antigens. That's positive selection, right? Sir? Yep. So the thing about this, when you're producing antibodies and so forth, at peak infection, you're producing to the tune of 2,000 antibodies per second. Okay? So 2,000 antibodies per second, there's going to be mistakes that will happen. So some cells may survive that, or antibodies survive that will attach to host cells. Okay? So those are eliminated, that's negative selection. And then you select for the ones that are going to react against foreign antigens. Okay? So you can see the difference between the two, right? Usually these are cloned to prevent all these problems of creating some cells that might attack a body cell. So when you have a cell that is sure to attack foreign antigens, what do? You make clones of it. You make a whole bunch of clones, which will be the exact same cell. So there will be no chance of what? Of making a mistake again and having a cell, creating a cell that will. So that's how the cells then multiply, by cloning. Okay? So cloning is a way of ensuring that you have a cell that you know is going to react against foreign antigens. And so you clone it. You just make an identical copy of it and multiply it many times. All right? So, B and T cells, of course, they will then migrate to the lymphatic system, right? We did the lymphatic system already, and they will be circulating in there. Okay? B cells come straight mature from bone, and then T cells pre-T cells will go to the thymus, mature there, and then once they mature, they will still circulate in the lymphatic tissue. All right? All right, so note the difference between the positive selection and the negative selection, okay? And the importance of cloning, because cloning ensures that you're making an identical copy of an assured cell that's going to react against foreign antigens. Okay, so cloning is important. So that's why when you have monoclonal antibodies, you usually have one type of antibody that will launch an immune attack against a microorganism. But then there are also polyclonal antibodies where you have several antibodies that will attack one, which is even better, because if one fails, there's always another one that will attack it at a different spot. Okay? All right, so that's that. Anyway, so we know about how the B cells mature in bone and then migrate to lymphatic tissue, and then the pre-T cells will go to the thymus, and then after that, they will still migrate to lymphatic tissue. All right. I just want to introduce right now, I just talked about how an antigen is a very long protein, or part of an organism, that will stimulate immune response. And I said it's 10,000, up to 10,000 in molecular weight. So that's a big protein chain right there. What the immune system does, because the antigens are big, they will react to a certain part, to a portion of that antigen, okay? Because it's just too big, okay? Which is called the epitope, or the antigenic determinant, and I'll talk about this later, okay? Antigenic determinant, okay? because it's just too big of a molecule for the whole, for the body to react to the whole thing. So those are called epitopes. Those parts that actually they will stimulate the immune response, a certain part of an antigen. Okay, and it's called the epitope or antigen determinant. And I'll talk about it also in a little bit. All right, so these are the molecules I was talking about. The major histocompatibility 1 and major histocompatibility complex 2. All right, so we talked about how big the antigen is, and we talked about how you have the epitope, okay, which is that part of the whole antigen that the immune system reacts to, and that part is called the antigenic determinant. All right, because they are so big. So there will be an antigen receptor on the T cells or the B cells, all right, on the surface of those lymphocytes. Usually the antigen receptor on the B cells is an antibody. So B cells, you have antibodies like that, okay? And they act as receptors, okay, on the surface of the B cell. And we'll talk about the different kinds of antibodies, immunoglobulins. So IgD and IgM will be located on the B cells as receptors for the antigen, okay? So that antigen receptor will combine with it will recognize the epitope, okay? So it's like an enzyme lock and key. So it only has to recognize the epitope, right? So I kind of introduced this earlier on. Major histocompatibility complex 2 are proteins that the body uses to exhibit antigens on the cell surface, okay? So let's say a cell is infected by any kind of cell. That's why major histocompatibility complex will exhibit antigens of any cell with a nucleus or nucleated cells. All right? So it could be any cell. It's not a professional immune system cell. It could be any cell that is infected, and then it will process that antigen, okay, and then it will go into the rhabdomyoplasmic reticulum, and then it will go into the Golgi body, where it is packaged in a vesicle, and then combined with MHC1. Okay, so that is an internal antigen, or an internal when a cell is infected. Okay, when a cell is actually infected, and then it will exhibit that antigen using MHC1. Okay, and MHC1 tells the immune system to destroy that cell. So it will stimulate cytotoxic T cells because that cell is infected. You see what I'm saying? It has been infected. So any body cell, like if you get a virus that comes in and then attacks that cell will process its, break it down, process into an antigen, combine it with MHC1 and put it on the outside of the cell so that it will alert the immune system to kill that cell because it's infected. Okay, so that's MHC1. And this will be important when we start to talk about activation of helper T cells, activation of B cells, and formation of antibodies. So that's going to be important. So MHC class 2 is usually used by the professional immune cells, like antigen-presenting cells, like dendritic cells, macrophages, B cells. Okay, when they engulf an external antigen, okay, because they will recognize foreign antigens, so they will engulf it and then process it. It doesn't go into the endoplasmic reticulum anymore. It's packaged into vesicles and it's isolated because the body already knows that it's foreign. Okay, the only one that gets included in the endoplasmic reticulum and so forth is one that infects the cell. So the cell kind of has no defense to know that, okay, this is not mine. So it's processed that way. Okay. Because even self-antigens are also processed the same way and are exhibited on MHC1, but that doesn't stimulate an immune response. Okay? So this is professional antigen-presenting cells, like B cells, macrophages, okay, and they will exhibit it on MHC2. When an MHC2 is exhibiting an antigen on the cell surface, the immune system knows not to kill that cell because it knows that it is one of the immune cells. So all it does, it will launch, activation of other cells, okay, and formation of antibodies and so forth, okay? So that's the difference between these two guys. MHC1, I need you to know, how one is processed, and I'll show you in a little bit, and how MHC2 is processed in terms of how it will exhibit the. So let's play this little. There are some cartoons that I want to play, but I'll have you play later on, just so that you can sort of go through this.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants, or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
Okay, so that's why I was saying earlier on, if you have different antibodies that will react to the same bug, to the same, foreign body, then you have a better chance, because if this one fails, then the blue one will get it. But there are certain times when you only have that one antibody that will attach to the epitope on that foreign microorganism. And then if it fails, then you start to have problems. So a polyclonal response is better than a monoclonal response. See what I'm saying? Okay, so this is what I was talking about. The MHC1, because it simply exhibits antigens of nucleated cell, all the cells in the body, whether it be even a professional cell, like a B cell, or any cell, or any cell that's infected. So what it does is that it will, process that internal. So this will be an internal. It's already in the body. Like when it infects, So it is processed. You see, this is a huge antigen. It is processed. So it's kind of broken down to smaller molecules that can be handled. So that's how it's processed. So it will then go to the, once it's broken down and processed into smaller antigens, then it goes into the rough endoplasmic reticulum, okay, where it will mix with MHC1, okay? So it will mix with MHC1, and so it will form the MHC1 antigen complex, all right? Then it goes into the Golgi body, right? We all know about the functions of these, organelles, right? So what does the endoplasmic reticulum, the Golgi body do? It will take that MHC1 antigen complex and bag it into a vesicle, right? And then once it's bagged into a vesicle, that vesicle, okay, is going to be, it will be part of the membrane, right? You can see how it's attached to the membrane, like that. So you have that, and then the MHC complex, and then the antigen, MHC complex. So when exocytosis occurs, this becomes part of the membrane of the cell, and then it will expose the antigen on the cell surface attached to MHC1, right? If it's a foreign antigen, then it will stimulate the immune system to kill that cell, right? It will stimulate cytotoxic T cells and so forth, and that cell is killed because it's infected. But also the body, the way the body can tell that this is foreign and this is not, is that it will show itself too. It will exhibit itself on the outside of cells by exhibiting its own self-antigen. So you say, hey, it's me. You know what I'm saying? Just like if you see somebody in the dark and they don't identify themselves, it could be your son. It actually happened sometimes. They thought somebody was an intruder, and it was their son coming in late, and they shot him, you know, because he didn't identify himself. So the body has to do that. It has to exhibit its own antigens on the cell surface and say, hey, it's me, so that it's not attacked by the immune system. So when that happens, this is the sixth part, no immune response is launched, okay? Because the body still has to do that. So that's why the processing of it goes from, in the cytosol, it's broken down into smaller antigens, and then it's packaged into a vesicle that goes into the rough endoplasmic reticulum, where in the rough endoplasmic reticulum, it gets mixed with what? It gets attached to the MHC1, all right? And then the MHC1 antigen complex will leave and go to the Golgi body, and then the Golgi body will package that vesicle of MHC1 antigen complex, and then it is exposed via exocytosis. And then if it's foreign, immune system launches the response. If it's not, then the immune system doesn't. All right. So now the MHC2. We talked about how B cells, macrophages, dendritic cells will recognize foreign, antigens attacking the body. So they will do what now? They will engulf it, break it down. So this is an external. That's number 2, the difference between MHC1 and MHC2, right? So that's an external foreign antigen that is engulfed, all right? When it is engulfed, you see here, this is endocytosis occurs, and it's engulfed, and then, it's mixed with lysosomes and vesicles and so forth. That will start to break it down, right? So it's going to be broken down here, and then that's being processed, okay? And then when it's broken down, because the body knows it's a foreign thing, it's not going to take it all over the place. It will immediately sort of suction it, like quarantine it in a vesicle. You know what I'm saying? Because the body is not going to take it to all the organelles of the cell, and then you have a problem there. So it is not going to take it to the rough endoplasmic reticulum. It takes it straight to the, to the Golgi body. And it's a, the Golgi body takes in vesicles that have the MHC2 in them. So it will be like a bag, pretty much. That's how the cell, moves stuff in vesicles, right? Little membrane bags, if you will. And then it fuses with that. And then, of course, this also fuses with that. This will be a vesicle containing the broken down or processed antigen. Okay? And then it fuses with that, all right? So you got endocytosis here of the molecule. And then 2, you process it and you break it down, all right? And then 3, the vesicles carrying the MHC2 and the vesicles carrying the processed antigen will, unite with this and double the contents into the Golgi body, all right? And then the Golgi body attaches MHC2 to that processed antigen, and then it is exocytosis occurs, and then what happens? This is a processed antigen on MHC2, and then it's going to stimulate formation of cytokines and so forth and other things that will then recognize that antigen on other cells and start to kill it. All right? But the cell itself that has done the presenting is not killed because it's a professional, antigen presenting cells. But there are times when these professional cells, because they are cells after all, get infected too. All right? And if that happens, then it is exposed on the MHC1 and is destroyed. You see what I'm saying? So MHC1 and 2 is a way to show the immune system to either destroy that cell or to make cells, antibodies, and so forth that will then deal with the microorganism. And if that cell is spared, the one that's because it's doing the presenting, right? It's a professional cell that presents antigens to the immune system to activate the immune system, right? You see the difference between MHC1 and MHC2? Okay, in the exam, if I ask how does the body process external antigens or internal antigens, or if I say MHC1 and MHC2, how is that processed? And then you'll be able to tell me, right?
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class I MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class I MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles, such as viruses and bacteria, by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class II MHCs, and the two structures combine. The MHC class II antigen complex is transported to the plasma membrane. The displayed MHC class II antigen complex can stimulate other immune system cells to respond to the antigen.
So you see the difference now? If you want to see, this is a good thing to listen to, and then it will show you. Because in the exam, I want you to explain how the MHC is processed, 1 and the MHC2. See what I'm saying? All right, very good. Okay. So, adaptive immunity. So, here we're going to be talking about now formation of helper T cells, formation of B cells, activation, and then, those cytotoxic T cells. Okay? So, MHC antigen complex, right? And there's usually co-stimulation. So, what happens is that when those cells recognize, the immune cells recognize the antigen presented on the MHC complex, there are other proteins on those immune system cells that will join with the other proteins on the the presenting cell, or the antigen, or the cell that's infected. Okay, the B7 and CD28 proteins. The CD28 proteins are on the surface of immune cells, like helper T cells and so forth, and the B7 is on the antigen presenting cell, or the cell that is being infected. So that also co-stimulates to produce some cytokines like interleukin-2 and interleukin-4. Okay, interleukin-4 is for B cells. Interleukin-2 will stimulate helper T cells to become active. Okay? So that's where the co-stimulation part, and I'll show you the proteins involved. And so the co-stimulation usually involves the B7, CD28 proteins. The CD28 on the immune cell and the B7 on the antigen-presenting cell, or the cell that's infected. Okay, and that will produce the cytokines like interleukin-2. It activates other immune cells. All right. Helper T cells do what the name say. They help other formation of more cytokines in a kind of sort of increase inflammation, and sort of help the T cells to become activated as well, the cytotoxic T cells. Okay? So antigen-presenting cells will stimulate proliferation of helper T cells, for that particular organism, and then those helper T cells will stimulate cytotoxic T cells and so forth, right, to kill that microorganism, right? And I'll show you. So this diagram shows the activation of helper T cells. Okay, so this is a professional antigen-presenting cell. It is engulfed an antigen and processed it via that way, that goes straight to the Golgi body, and it has exposed it on the MHC class 2, right? Because if it's an external antigen and it's engulfed, it goes to MHC class 2. Because MHC class 2 is one that will stimulate other immune system cells to launch an attack. Okay? Okay, so the helper T cells will recognize, this is the T cell receptor, which is usually an antibody, right? It will recognize that antigen exposed by the MHC complex 2, right? And then, of course, the T cell receptor, okay, on the helper T cell will attach to the processed antigen exposed by the MHC class 2 proteins, right? So, in this case, you have this protein here, CD4, okay, always attaches to MHC class 2. That's how the body knows that it is not MHC class 1. See what I'm saying? Because otherwise the body doesn't know that this is MHC class 1 or MHC class 2. Okay? So the MHC class 1, they attach to CD8. Okay? So CD4 is MHC class 2. So the body knows, okay, we're not going to be killing this cell, we're just going to be doing other things that will then launch an immune attack against that microorganism. You see the difference now? Okay? If I ask about activation of helper T cells, this is what you talk about, right? And usually it's a macrophage or a dendritic cell that has engulfed a bacteria, you know, or a foreign microorganism, has processed it, and has exposed it on the MHC class 2, okay? And then the helper T cell receptors will recognize it and attach to it, okay? And then the body knows that it's an MHC class 2 because it will fit to the CD4 proteins. Okay, so the CD4 protein on the helper T cell will attach to the MHC class 2. Okay, so now this is the co-stimulation I was talking about. The B7 is going to be on the presenting cell. You see this B7 right there? It's going to be on the presenting cell. And then the CD28 is going to be on the helper T cell. So that helps to co-stimulate and start to produce cytokines. It kind of helps to stimulate, because this is a naive T cell. You see what I'm saying? What does naive mean?
Mature.
Yeah, in naive, it doesn't know about that bug. Because we're trying to create helper T cells that will recognize that, okay? So it's naive right now because it hasn't met that bug before. So it is co-stimulated by the B7 and CD28, all right? After that co-stimulation and all that activation, the helper T cell is now activated, it's excited now. It will start to produce interleukin-2 receptors, okay? Then it will also produce interleukin-2 itself, okay? And then the interleukin-2 will attach to the interleukin-2 receptors that have just been formed, all right? And then once that happens, it will stimulate, okay, the cell to then divide and differentiate. Okay. So you will produce memory T cells that will recognize that specific bug and they go into storage. Wait for the secondary response. If you are ever exposed again, so that's why when you get the flu for the first time, you get it and you become sick. But if you're exposed again to the same flu, to the same virus, nothing happens. Okay? Because of this, they produce long-lasting. They can last up to 10 years in your system, you know. So they last almost a lifetime. So your body will always be able to recognize it. And then it produces, this is no longer a naive T cell. This is now competent against that particular microorganism, though, because this is specific, right? Only against this antigen for this specific microorganism. So then this will produce cytokines that will regulate its process number 8. It will produce cytokines that will then recognize and stimulate the production of that helper T cell that will immediately attack the bug. or it will immediately, stimulate other cells like cytotoxic T cells that will kill that specific bug. Or it will stimulate B cells that will produce antibodies against that specific bug or that specific foreign microorganism, right? So that's the activation of helper T cells. Because the helper T cells are the first cells, that will be attacked. Especially in HIV, it's CD4 positive T cells that are attacked by the HIV virus. Okay? And there will be those naive T cells. All right, so that's the whole process right there. So this is the process of activation of T cells, helper T cells via the antigen-presenting cells, like dendritic cells. Sometimes it's B cells, sometimes it is macrophages. Okay, and it involves co-stimulation of CD28 and B7 on the antigen-presenting cells, and it produces interleukin-2. Interleukin-2 is the final act that will stimulate the division into the memory T cells and helper T cells. So the process is going to be the exact same thing, except we'll just be dealing with different proteins the MHC one, okay? Because the MHC1 kills those. So this is the T cell dependent. Okay.
Antigens, such as bacteria or viruses, are usually ingested and degraded into small fragments by phagocytes, such as macrophages. The small fragments are then complexed with a special type of protein called a class II major histocompatibility complex protein, MHC, and transferred to the surface of the phagocytic cell. Helper T cells recognize an antigen when it is presented on a class II MHC because helper T cells have special recognition proteins on their surfaces called T cell receptors. This interaction activates the helper T cells. The activated helper T cell is now capable of stimulating B cells to become antibody-producing plasma cells.
So now I'm going to show you how those helper T cells help to activate the B cells now. So we're going to talk about B cell activation. Same kind of process that we talked about, but now the helper T cell that's just been activated is now going to activate the B cells, right? All right, so before we go into that, we'll talk about tolerance, because that's how the body will not attack its own antigens, all right? So pretty much tolerance is suppression of an immune attack against a particular antigen, all right? So our body will tolerate its own cells, right? Because it doesn't attack our own cells, unless you have a problem where you have an autoimmune situation where the body will start to attack its own cells. So that's something screwy has gone on with the tolerance, okay? And what does tolerance, how is it produced? Of course, it will delete self-reactive cells. Remember, we talked about how we have the positive selection and the negative selection, right? So you still remember that. So it prevents lymphocyte from then becoming activated and attacking that antigen, right? So they also suppress the T cells. Right now we just talked about helper T cells that help to excite the immune system, right? They are also suppressor T cells that will suppress it, right? And there's two kinds of tolerance. Central tolerance. Central tolerance happens when they are being produced, like in central, lymphatic tissue, like the bone marrow and the thymus, right? So in that case, when you're producing, remember I was checking how this body is producing the cells at a record speed, right? So it's going to make some mistakes. So just like any assembly line, like we always say, nothing is ever 100% efficient, okay? So it's going to make mistakes, but the body will recognize those cells that might attack self-antigens and it will destroy them. Okay, so that's negative selection, right? So that when that's occurring in the central, during production, like in the thymus where they mature, or in the bone marrow, right? Red bone marrow, that is central. Okay. Then let's say this cell has escaped central tolerance screening methods, and then it becomes a cell and it becomes a mature cell, there is what is called peripheral tolerance, right? Peripheral tolerance will then deal with those cells that have survived the initial checkpoint, right? In the periphery, like in the lymph nodes and all this peripheral lymphatic tissue, right? Do you see the difference between the two now? The central tolerance occurs during production, and then if any escapes, peripheral tolerance will take care of it and will destroy those particular cells, immune cells that will launch an immune attack against self-antigens, right? Okay, so gotta know these two, right? Okay, so we have activated our helper T cell, right? Okay, and now we have, it's gonna activate the B cell to produce what? Antibodies against that specific microorganism, right? There are very few instances when once a B cell attaches to, a B cell receptor attaches to a foreign antigen, it immediately goes to step 6, where it will start to produce the memory B cells. There are some antigens that are like that. It skips all these steps, but very few though. Most of them require the activation from helper T cells. Remember, we just activated the helper T cell, and we now know exactly what we're dealing with. So the B cell will then become, first it has to attach, because remember the B cell has got antibodies which are receptors that are set on the cell surface. All right? So it will attach to this, and this is unprocessed, to the antigen, right? And then, remember the receptor-mediated endocytosis we talked about, where whatever is being engulfed attaches to a receptor first. So that's what occurs here. Receptor-mediated endocytosis will engulf this part, and then it is internalized in a vesicle. Right? Remember those transport mechanisms we did in NP1? Okay, so this is, of course, has been processed, right? And it's now exposed on the, as long as the bug is coming from outside, the antigen is external, it's going to be processed and exposed on MHC class 2, right? Okay, so MHC class 2, and then the helper T cell will come and attach to that. So the T cell receptor, okay, will attach to the exposed, antigen on the MHC class 2, because it was an external one. It was engulfed via receptor-mediated endocytosis, right? And then it will attach to that. And then, of course, CD4 always attaches to MHC class 2. That's how the body knows it's MHC class 2. Okay, so CD4 attaches, all right? And then it's, because it's already stimulated, right? This is already stimulated, so it's going to start to produce interleukin-4 now, not 2. That was for helper T cells. This is the same process, though. It's just different proteins here. So we produce interleukin-4, and then the B cell will, will then produce those interleukin-4 receptors, and this will attach there, right? So now that the B cell is now activated and it's all excited, it will produce memory B cells and plasma cells. Remember, the plasma cell is the one that actually produces the antibodies. And this one will stay for memory, right? So activated B cell produces memory B cell and plasma cell. And the plasma cell will produce a whole bunch of antibodies. Like a lot. And remember, at peak infection, I was telling you that the body will be producing 2,000 antibodies per second. That's amazing. Because an antibody is a long protein. And you're producing 2,000 of those long protein molecules in 1 second. Can you imagine that? If you wrap around your brain around that, that's amazing. All right? And then it will produce a whole bunch of these. Antibodies will be for that particular, because this is adaptive immunity, and it will recognize that particular antibody. Right? So the process is the same for activating B cells, right? And you get this. So it's nothing different now. It's nothing new except that we have a few different proteins, and that's all. Right? That's it. So once you learn the process, you just know that the B cells, the interleukins involved are 2, and for helper T cells, are 4, interleukin 4, and helper T cells is interleukin 2. The rest of the things are the same. You produce memory B cells, and plasma cells that are producing the antibodies, or with the helper cell you produce memory T cell, memory helpers T cells, and then of course the helper T cell that will recognize that specific part. Okay, so that's the whole process. Okay, except for this B cell activation, we are trying to also produce plasma cells that produce antibodies, and these antibodies will attach to that specific bug, and I'll show you how the antibody will deactivate or kill the antigen in a little bit. Now that we got a whole bunch of antibodies from the B, right? The B cells. So, antibody-mediated immunity with the B cells. We've activated B cells, we produce plasma cells, and so we have this. So pretty much this is like an immunoglobulin, which has got kind of like a Y shape to it, right? And on this bit here, this is the variable region. Okay, this is the variable region, and this here, and part of that is the constant region. So that means all the antibodies ever produced, the constant region is the exact same sequence of amino acids. Okay. Now this is the part that attaches to the antigen. Okay, the variable. Why would this part then be variable and this part be constant? Why is it necessary that this part that attaches to the antigen is variable? It's not a trick question.
So it can bind
To what? You're on the right track.
antigen.
Yeah, and the antigens are? How many are there? It's like a million of them. A lot. So you're going to need different kinds of this bit of the molecule. So that's why that is variable. You see what I'm saying? And the rest of it is constant, so that when the body is making, it doesn't have to make a whole new antibody with the whole. That's why it can make how many? 2,000 per second when it at peak infection, because the big part of the molecule is constant. It's the same amino acid sequence. Only the part that attaches to the bug or to the antigen is going to be different, because it's going to be different little antigens.
So the just.
So this is the only part that is variable, that will differ in amino acids, because it will be attaching to different antigens. Okay? You see, it makes sense, right?
Yeah.
Yeah, it does make sense. Anything the body does make sense. So if I ask about the variable region and the constant region, you know the significance of that, right? The constant region is the bigger part of the molecule, and the variable region is the smaller part on the ends. That is variable because it's the one that attaches to the different antigens. So it has to be different, okay? Because otherwise it cannot attach to different antigens, if it's going to be the same region. Okay, so that makes sense. Anyway, there are also certain parts on the constant region that will attach to complements. Remember those complements? Those proteins that I say, they also activate all these antibodies so that they attack the site of binding for macrophages and basophils, and mast cells. Is going to be this part here, because basophils and mast cells will produce inflammation, and then it will attach onto those antibodies attached to the B cell, to the mast cell, like at this point. Okay, so the parts of the constant region attached to complement, and the other parts attached to the mast cells and basophils. Okay, so remember I told you the different kinds of immunoglobulins. Most of them are IgG, and they activate complement, all right? And they can also pass through the placenta to the fetus, okay? And these are the ones that are responsible for hemolytic disease of the newborn, because they are transferred to the, and also responsible for the resus reaction, that gives that hemolytic disease, right? So, so if you want to know it's gamed or you've been played, right? G-A-M-E-D, kind of roughly in that order, all right? In terms of, how much there is of that. So most of them are the IgG. And then IgM is one that is a B cell receptor protein, okay? It's responsible for the blood transfusion reactions, okay? It also activates complement, by the way, okay? It does that, okay? And it's the B cell receptor that is responsible for the ABO transfusion reactions, okay? So the IgA, it is secreted in saliva, in tears, and any of the mucous membranes. It's what gives kind of defense to the body. For anything that takes external, like the digestive system is lined by the IgA. Is one that is the immune system, immunoglobulin that is responsible for protecting the body from things that are coming externally through any, or even the respiratory system, any mucous membrane, saliva, tears, all those have IgA. Sometimes I ask you about the 5 different immunoglobulins, and I'd like you to tell me about what they do, right? What's their function, okay? And then, of course, IgE is usually involved in allergic reactions and launches inflammation. It attaches to the mast cells and basophils. Remember that part I was showing you on the constant region of the immunoglobulin, of the that attaches to the, so it will be the IgE. IgD is also another one that acts as a B cell receptor, okay? But it's not involved in the blood transfusion reactions. The one that is involved in blood transfusion reactions is IgA. So that's how you can differentiate between the two. If I ever ask about you to tell me about the 5 immunoglobulins and their functions, you would have to distinguish between those two. Don't just say they are both B cell receptors, but then you gotta tell me something to show, to differentiate them. Okay.
So IgA is the reason your blood clots when you get transfusion, and it's not the right kind of blood.
IgM.
Oh, it's IgM.
IgA is in saliva, tears, and stuff like that. Yeah, and the mucous membranes. Okay, so if you want to remember them, GAMED, okay, G-A-M-E-D. Okay, so this one is more numerous though than the E. See what I'm saying? That's the only thing. Otherwise, this is the order in which they are numerous. Okay, going in descending order. You see that? Except these two. You see that? So you know. So gamed. So this is how, once you produce a ton of those antibodies, this is how they're going to inactivate. So they can interfere with the antigen by binding to it and interfere with these processes. Okay. They can precipitate them. So that means one antibody, because the antibody constant region attaches to itself. Okay, so the antigen is attached here, and then the Y will attach together like that, and that's precipitation. Okay, one of them is just binding to them and interfering with their processes, and the other one is actually precipitating them out of solution, so they cannot react. Right? What's the other one? It increases phagocytosis. This antibody attached to the bag will go and attach to the macrophages, and then it's killed. Okay, then it makes it easy for phagocytosis to occur because it will just go to the macrophage to kill it. So increases phagocytosis by binding to macrophages, and can also activate complement. Remember that part I showed you on the constant region where complement attaches, okay, the complement proteins, and then via the classical pathway is the one that's called that activates the complement that way. That's the classical pathway where you use the immunoglobulins.
Complement attaches to constant.
To the constant region, yeah. Remember, kind of here, you see, that's where it attaches.
Oh.
Yeah, and then it will activate complement. Remember what I said, complement is a group of proteins that can insert themselves, make pores into the cells that are infected with the bugs, and then they pretty much burst. Okay? Because it inserts big holes. All right? And then it can also attach to mast. Remember, the mast cells and basophils, they attach down here on the bottom part of the complex, on the constant region, and it will activate, because basophils and mast cells will release inflammatory mediators that will cause inflammation. What does inflammation do? Pretty much blood rushes there, carrying a whole bunch of white blood cells and so forth, right? So that will help to kill the bug. So sometimes I ask about how the antibodies will disable the antigen. That's how it is, those 5 ways. Okay? Attaching to them and disabling their processes, you know, and just pretty much precipitating them, macrophages activated, and antibodies. So these are them. Okay? All those processes. Right? So you just know how the antibodies deactivate or kill the antigen. Alright? Alright, so that's what we talked about, right? Because of those memory cells, we have a weak first response because the body is still naive, it doesn't know the microorganism. And then the secondary response, upon secondary infection, you launch a more robust response. Why? Because you already have these memory cells lying and waiting, right? So we know about that. This is because of the memory cells. Okay. Now this shows you a B lymphocyte, a B cell. Do you see? Last time the confusion I had was, you see how thin the cytoplasm rim is. See how big the nucleus is on this one. So you can see this is a lymphocyte for sure. The other lymphocyte that was shown in the diagram had a very small nucleus and it had too much cytoplasm on it. And so that's why it tricked me. Anyway, but now, because I want you to see the difference between a B cell and a plasma cell. Okay, you see how it's got a big nucleus and then of course the nucleolus and then the microvilli a little bit. Okay, now look at the plasma cell. See the difference? What's the difference with the plasma cell and
A lot more cytoplasm.
a lot more cytoplasm, and you got all these mitochondria and so forth. That's what I'm saying. The other one, it's all nucleus. Now a plasma cell, because it's one that's going to be producing a whole bunch of these proteins and antibodies, it's got a full complement of organelles for that. You see what I'm saying? Makes sense? So that's what the B cells will differentiate into the plasma cells, okay? And this is just showing you the primary response. You produce fewer memory cells, fewer B cells. And then the secondary response, because your body is no longer naive, it knows it, it will produce much more B cells and much more, and a little bit of memory cells, because you know, you want to keep the thing going.
What makes B cells turn into plasma cells?
That activation, remember we talked about it. Yeah, when B cells come across antigens, what they do, they will engulf it, right, via that receptor-mediated endocytosis, right? And then it will be exposed on it, on MHC2. And then the helper T cells recognize that. And then they become activated. When they're activated, they will differentiate into memory cells and plasma cells. And then the plasma cells produce a whole bunch of antibodies. So we're going to skip this because it's pretty much talking about how these, antibodies will attach to these different epitopes, right? So you can watch this on your own. So we watched the important ones. This one it's important to, but it's pretty much talking about things we've kind of been talking about all up to now. So now we are left with, stimulating cytotoxic T cells in much the same way as we stimulated helper T cells, as we stimulated B cells. Okay. So this one is going to be the same as like the helper T cells. Okay. So now we're forming cytotoxic T cells, ones that will kill that cell. So now we're talking MHC complex 1, because the cytotoxic T cell will kill that cell. Okay, so we already know about this, right? And then of course it will release cytokines, from the helper T cells, and then the helper T cells will excite a whole bunch of these cytotoxic T cells. Okay, and they will be killing a specific bug for a specific antigen. Okay, they'll be killing cells that are infected by that specific antigen, cytotoxic T cells. Okay, cytotoxic T cells usually kill the viruses, like the T cells. That's why when HIV attacks the body, it will disable the CD4 positive T cells. Okay?
So MHC class 1 deals with viruses.
Yeah, or any external antigen. Okay. any internal antigen that comes into and infects the cell. Sorry, any internal antigen that infects the cell will be exposed on MHC1. Okay, because once it's inside the cell, it has infected that cell, and you need to kill that cell. Because if you don't kill that cell, what's going to happen with the cell? It's going to start producing all these viral proteins and so forth, and because the virus cannot reproduce by itself. So what it does, it will go into our body cells and use our machinery to make its own protein. You know what I'm saying? And that's not good for us. So what you have to do is kill the cells that have been infected, and they will be attached to the MHC1 when they explode. So same process again, guys. This is a dendritic cell. It is attached to a virus or an antigen that is on the MHC class 2 here for the helper T cells. Because what's going to happen is that some of them will be externally, engulfed, okay? Because these are, and some of them will infect the cells, okay? So the internal ones will be processed this way, okay, with the MHC class 1, okay? Because these things will be happening kind of simultaneously, you know what I'm saying? It's not like this happens and then, it's not like that. Even though we're breaking down into steps like this. So we've already done the MHC class 2, we know about that, right? And it attaches when a microorganism is external engulfed, okay? So here, the MHC that is bound and is internally processed, okay, is going to be exposed on the MHC1, all right? And then the MHC1 will bind to the cytotoxic T cell because it's going to be killed. But this one right here will bind to the helper T cell if it has been externally, you know what I'm saying? If it has been externally acquired and then broken down and processed via those ways, okay? So it's either or, you know what I'm saying? It's not like sometimes the cell is infected, or sometimes the cell engulfs it and brings it. So I'm just showing you the two. So that's why here it's either infected or through phagocytosis. You see what I'm saying? So I'm just showing you the two things simultaneously at the same time. But it's either or. All right? And then, like I say, MHC1, you have the, the CD8. Okay? MHC class 2 is CD4. Okay? But it's the same process like we talked about. No different. Okay? And we still get the same co-stimulation, right? With the B7 on the antigen presenting cell and the CD28 on the cytotoxic T cell. All right? So then it's going to become activated, and the cytotoxic T cell produces the interleukin 2. The helper T cell produces the interleukin 2. Okay, or if it has been internally processed and the cytotoxic T cell is attached, then it will produce receptors for interleukin 2 and interleukin 2, and then it will attach here and it becomes excited. Also, a helper T cell will attach here and do that, and then it will produce these interleukin 2 that will then excite other cytotoxic T cells, and then they'll come and bind to whatever cell is infected. Okay? Because it will recognize the MHC1. And then those cells are killed. Okay? All right. So interleukin 2, of course, will stimulate cytotoxic T cells to produce memory T cells and competent cytotoxic T cells. All right? Okay? And then, of course, the memory T cells produce the long-lasting immunity. The cytotoxic T cells, okay, will start to kill. They'll be competent, and they know that part. So they'll start to actually lyse any cell that is infected, because it will be exhibiting the antigen on the MHC class 1. Okay? So this process is not different from, so you just have to learn the first thing. Okay? And that's it, you're done. It's not a new thing here, and then of course the cytotoxic T cell that is competent now will be starting to kill any cell that's infected. And then, of course, the helper T cell, assists these ones, okay, to activate more cytotoxic T cells. Because once the helper T cells are activated, they'll go to the B cells, right? And they'll do that, or dendritic cell, whichever presenting cell it is, you know what I'm saying? And then it will cause it to form all those other cytotoxic T cells. So helper T cells will stimulate, also is increased because it will form a bunch of this B7. The B7 will attach to CD28 for co-stimulation. So you form more, okay? So this is the same deal, right? Okay, same process. Just know that internally processed antigens are exhibited on MHC1. Externally processed are exhibited on MHC2, and that's it, man. It's the same process. And then you form memory T cells and competent T cells, whether it be helper cells or cytotoxic T cells, same thing. The only difference is the B cells will differentiate into plasma cells that produces antibodies, and that's it. Okay, so that's the same process that shows activation right there, right? And then we're going to skip through this because this is just showing how a virus infects a cell, and then it's going to be exposed on the MHC class 1, right? And then you can watch this cartoon later, all right? And then, of course, the innate immunity, adaptive immunity, all those processes, guys, they're not happening like one after the next. They're happening at the same time. The body does it all at the same time. It's just that for purposes of learning it, we split it into the innate immunity and then we split it into the adaptive immunity. All right? But it's all happening all simultaneously at the same time. Okay? Activation of these macrophages and inflammation, adaptive immunity, antibodies, cytotoxic T cells, all that. Helper T cells at the same time. All right? And then the last bit is acquired immunity. It can be active or natural or passive. Active, your body is actually producing the antibodies. And passive, your body is not producing the antibodies. You are just giving it already processed antibodies, already made. Okay?
It's like a shot.
Yeah, you get a shot. So the active, when your body produces, it can happen two ways. When you're naturally infected, like if you get a flu and you get sick, that's active and it's natural. And you actually get sick and natural. Or we can give you a killed virus and so forth artificially via vaccination, and and then you start to produce actively the antibodies. But we give you an attenuated virus or attenuated bug, so you don't get sick. Okay? And then of course for passive, natural is through the mom. We said the IgG passes through the placenta, and then of course through milk, colostrum. The first milk has got antibodies, so that's passive. You're already getting ready-made. And artificial, you can inject, like if you get bitten by a rabid dog, what happens? They give you like injections of antibodies over a long time. Okay, because they'll be giving you a protein. You can't take it. Also, why? Because you will do what? You will digest it. No, your body will digest because it's a protein. Remember, an antibody is a protein, so your body will digest. You get amino acids, and that won't help you. So what you need to do is inject it, so you get it to artificially introduce it. All right. So just know the difference between this acquired immunity, the active and the passive, and the two methods: natural and artificial. All right. And then, as usual, we will always compare the two, the process, the systems, and so forth, and see how they affect you. Thank you, guys. I appreciate it. We'll see you in a little bit.